quickly moving to, that was sort of lung cancer without a specific marker, but looking at specific subsets of non-small cell lung cancer, EGFR mutations, I think, as many of you know, is a very common subset of lung cancer that has a genetic marker. And this ADC called patritumab deruxtecan, or HER3-DXd for short, was evaluated in patients who have EGFR mutant non-small cell lung cancer, had already received an EGFR targeting pill and chemotherapy. So their cancer had grown after a couple of lines of therapy. And basically, they picked the dose for this drug and looked to see what the response was. And here's another one of those waterfall plots. And I think, as you can see here, it's pretty fair. It's actually, decent performance here in terms of how well does it perform for these patients. But I think the takeaway message here from this sort of busy bars here under the waterfall is that when patients have cancer grow after after EGFR targeting therapy, there are many different reasons or mechanisms of resistance why that might happen. And this drug appeared to be active despite the mechanism of resistance. And I really think that's where its ability to shine lies, and that we might be able to reach for it in sort of a number of subsets of these patients with EGFR mutant lung cancer that has grown after an EGFR targeting pill. Coming up, there is a new trial which is going to look at HER3-DXd versus chemotherapy after EGFR TKI, that's tyrosine kinase inhibitor, or the pill. So for patients who have received targeted treatment with a pill, they're going to randomize patients 1-to-1 to this drug HER3-DXd versus chemotherapy. And I think that this trial will be very enlightening to determine, can it outperform chemotherapy in this setting, which I think is a very standard option that we often reach for. So that this will be exciting to sort of understand these results. One other subset of non-small cell lung cancer is MET positive lung cancer. MET is a protein that can be expressed on the surface of the cell. And so scientists thought that this was going to be a good target for an ADC, as we've sort of discussed this whole time, that the antibody can come and target the antigen on the outside of the tumor cell. So because MET is expressed outside the tumor cell, we thought that this would be a good option. This drug, telisotuzumab vedotin, targets MET and was tested in a number of subsets, but it found it was only really efficacious enough in patients who had non-squamous lung cancer and had EGFR wild type, so non-mutated cancer. And so patients received this drug in the later line, so they had already received, chemotherapies or immunotherapies. And the hope was basically that this biomarker, this target, can sort of serve as a proxy for seeing, like, how well might people respond. The CMET high, so it's patients whose tumors had a really high expression of this protein had a response rate of about 34%, intermediate 22%, and overall just about under 30%. I think that's fine, but I think overall we do sort of hope for more for that for our patients, especially with such a targeted therapy. So remains to be seen what will happen with this drug. I think something to consider and to look out for moving forward are going to be these randomized trials, which are currently enrolling, but we don't have any data for, are for looking at whether these drugs are going to replace any proportion of the standard of care, or if are they going to be used in combination with current standard of care, which is platinum-based chemotherapy and immunotherapy. If we were to combine these drugs, for example, ADC and chemo and immunotherapy, is that efficacy going to be very impressive and enough to make up for any sort of extra side effects or toxicity? I think these questions are going to be super important and remain to be seen. And the results of these trials will be able to tell us, for example, for people who have a PD-L1 score on their tumor, which is, a marker of how well immunotherapy alone might work. If they're randomized to immunotherapy alone versus This is datopotamab deruxtecan plus immunotherapy. That will sort of give us the data. How extra effective is it? How extra toxic is it? And so we really need to see these data to sort of understand how these drugs are going to fit in the future, and whether there might be special populations of patients who might stand to benefit most. For example, maybe patients with brain metastases might need this escalated therapy, or other sort of vulnerable subsets. This is just a quick slide to show you that while ADCs themselves are a new drug, there are many sort of even further novel developments moving forward with, all parts of the engineering of the ADC sort of under development, novel linkers, novel targets, novel payloads. And so all of that is coming, and mostly in preclinical development now, but will be coming to clinical trials soon in the coming years.